Hello students, in our previous lesson we discussed the process of gametogenesis, and I told you how the gametes actually form, what is spermatogenesis, what is oogenesis, how the egg and sperms are formed, and what is the difference between a primordial cell, that is a diploid cell, and how it is changed into a haploid cell. So the whole process we have to discuss in meiosis. Actually this process is going to be taking place at the back end of the process of spermatogenesis or oogenesis. But what happened actually? How the cell that is diploid in chromosome numbers will be converted or will be changed or shifted into a cell which is haploid in chromosome numbers. So let's discuss the process of meiosis. We'll talk about meiosis. In previous lesson we discussed this process of meiosis. It takes place at a time of gametes formation, during spermatogenesis or oogenesis. When we talk about the process of meiosis, two phases of meiosis actually are there: meiotic 1 division and meiotic 2 division. In first division, that is called meiotic 1, the parental cell that contains 46 number of chromosomes, the diploid number of chromosomes. We discuss the terminology diploid, haploid, what these terminologies are. Let me first tell you about the meiosis process, how this process is going to be taken place. In first step, during meiotic 1 division, one parental cell will be divided into two daughter cells. But when we compare it with mitosis, in these two cells the number of chromosomes becomes half. Now they are haploid numbers, 2n. Over here we will write 1n or simply n number of chromosomes. If 46 number of chromosomes are there in the parental cell, these two daughter cells will be having 23 number of chromosomes in both. Then these two cells will further be divided into 4 cells during the process of meiotic 2 division. This meiotic 2 division is similar to mitosis process. These 4 cells will be genetically identical to these 2 cells. Number of chromosomes will not be reduced further. If they are 23 after first meiotic division in these 2 cells, after meiotic 2 division in these 4 cells, again the number of chromosomes will remain constant to 23. So let's see. As previously we discussed the process of spermatogenesis, what happened over there, how this process of meiosis took place. We discussed its different phases, but let me tell you some very interesting information I want to share with you. We will talk about the spermatogonium cell, which enters in the process of spermatogenesis. This spermatogonium cell was a diploid cell, with 46 number of chromosomes. In this cell, actually, there is a combination of two different type of chromosomes. There are two sets of 23 chromosomes. The first set is from the paternal side, and second 23 set is from the maternal side. Like if in the testes of a person the process of spermatogenesis is going to be taken place, all the cells in the body of that person having two sets of chromosome, that person will receive one set from the father and the other set from the mother. So when the person will be having 46 number of chromosomes in all somatic cells or body cells, it means actually there are two different sets of chromosomes. I mean, these 46 chromosomes actually represent half number of chromosomes which the person will get from two parents. I mean, the sperm will carry 23 chromosomes, the egg or ovum will carry 23 chromosomes. During the process of fertilization, when the nuclei of these two gametes fuse together, the 23 number of chromosomes combines together, and 46 number of chromosomes will be restored again in the zygote. So during development, that zygote will be divided by mitosis, and in the whole body, all cells which carry 46 number of chromosomes, actually they are the mixture of these two sets, 23 from the father and 23 from the mother. So when we talk about the two types of chromosomes, I mean, this chromosome will represent a chromosome set from the father, and this will a chromosome from the mother. Okay, what happens next? Actually, when the process of meiosis will take place, we are having 46 number of chromosomes in primary spermatocyte. This number is called diploid number. Diploid number of chromosomes. That means one end from father and one end from mother. 1n plus 1n, that will be equal to 2n. So in both these conditions, actually every set represents half number of chromosomes. So when these two sets combine together, we will be having 46 number of chromosomes. So that set of chromosome now will be called diploid number of chromosomes. Diploid, that will be represented by 2n, I mean one from paternal side and one from maternal side. Another interesting information, when we talk about the structure of these chromosomes, actually these chromosomes, it's a chromosome and this is a chromatid. It's a single structured chromosome. A single chromatid is there which is attached by structure called centromere. But keep in mind, when the process of meiosis takes place, as we discussed in mitosis during interphase, during S phase, what happened over there? Duplication of genetic material. DNA was replicated. So over here, the process of replication will also take place. When the process of replication will take place, so let's see what will happen over there. This chromosome after duplication will change its face. Now it will be like this one. Right now, why we are having two different chromatids? The reason, this chromatid contains a specific amount of DNA in it. Every chromosome is made up of DNA, and there are certain positions on this chromosome called gene locus. What is a gene? unit of inheritance, a small length of DNA that carry messages which are actually transferred from parents to their offspring. So these are the locuses or gene loci. So in both these cases, when we talk about the chromosome from maternal side and paternal side, these chromosomes are called homologous chromosomes. We will discuss in the process of meiosis, when we will discuss the stages of or phases of meiosis. Over there I will tell you about the concept of homologous chromosomes. But homologous, when you talk about the homologous concept, keep in your mind, in one cell there are 46 number of chromosomes. If we have 46 number of chromosomes, it means we have 23 pairs, and in those 23 pairs, each pair will be having one maternal and one paternal chromosome. So 23 pair of chromosome will be there. So during the process of meiosis, when they arrange themselves during first phase, prophase, we will observe these two chromosomes, I mean one from the maternal and one from the paternal. Why they are called homologous? Actually, the reason, they find each other. Like this is set 1, it's set 2, then set 3. So in these 3 set of chromosomes, when actually they arrange during the process of meiosis, every chromosome of set 1 that belongs to paternal side will definitely find the chromosome number 1 of maternal side. How they find each other? It's a myth, but it's very interesting. I mean, when the process of meiosis starts, actually during that process, when these homologous chromosomes pair up, I mean number 1 chromosome of maternal side will find paternal side number 1 chromosome, number 2 maternal side will find number 2 of paternal side. So this is a way in which they arrange into 23 pairs. The first 22 are called autosomes, and last one is not a homology in males. Basically in males we are having XY chromosomes. So Y chromosome is shorter in length. Keep one thing in your mind when we talk about homologous pair of chromosome. Homologous pair of chromosomes are those chromosomes which are same in their size, shape, structure and amount of genetic material, along with position of centromere. So if they are similar in shape, size, structure, amount of genetic material, the amount of DNA, so they will be called homologous pair of chromosomes. But when we talk about the last pair, that is sex chromosomes, XX in females will be a pair of homologous chromosomes. But when we talk about the XY, it will not be a homologous chromosome, because Y is shorter in length. I mean, definitely it will not carry out some functions in coordination with X chromosomes. Okay, this was the concept. Actually we will discuss this concept further. I mean, why first they were single structured chromosomes, how they change into double structured chromosomes, why they were changed into chromosomes with 2 chromatids now, and how the amount of DNA was increased. Actually, here the amount of DNA is replicated from this one chromatid. Another chromatid is formed, mean the amount of DNA is formed. In this phase, it is called bivalent or tetrad. So these two chromatids, they are called sister chromatids because they are present on the same chromosome. But this chromatid, when we compare it with this one, it is called non-sister chromatid. Okay students, what happens next we will discuss in our next lesson. Till then, stay happy and blessed.